right, I'd really like to thank the organizers for inviting me. And first I'll see if I can figure out how to use this exciting device here. There we go. All right, it's live. So I'm going to talk to you about something very different today, and this is really about how to use computers in studying the microbiome. Just first, I'm a physician, so these are my disclosures, although there is no influence for this talk. So what am I going to talk about? I'm going to really start off with the history of artificial intelligence. I've been in the field for over 30 years and want to give you some perspective of where it's come from and where it's going. I'm then going to talk about sort of what's going on right now, the revolution in the field, and then switch to the challenges with the microbiome, but really some of the opportunities there. And then finally, I'm going to end with some cautions. So first off, the history of the field, it started really in the 1940s. This is a paper from 1942 with McCullough and Pitts. And what they were trying to do was model the human brain, viewing it kind of as electronic circuits. And so it was a set of mathematical equations called neural networks. So back in 1942. The field continued at really a good clip in the 1960s and 70s. Interestingly, one of the early applications was to microbiology. So this was a system developed at Stanford in 1975 called MITRE. And what it was a set of these rules that were developed by physicians and microbiologists. And it was trying to, you would tell it characteristics. Here's what a gram stain looked like. Here's a certain clinical characteristics. It would try and identify the organism and recommend a treatment. It actually did quite well, but it was not widely used because there were not lots of computers in hospitals in 1975. So this sort of gap between society and technology existed even then. At the same time as these sort of expert systems were being developed, there was another force going on in AI, which was called machine learning. And this term was first coined in the late 1950s. This is Arthur Samuel. He invented a program to play checkers. And the idea with machine learning, it was a very operational definition. It said the computer is going to get better at a certain task, given more data, more trials, et cetera. So a process called training. But it wasn't trying to mimic human intelligence, just to try and do better and better at a task. So what happened between then and now? Starting around 2010, there's what we call the deep learning revolution. And interestingly, this stems from a lot of the same tools that were developed in 1942 for those neural networks I mentioned. The big differences are a few things. First of all, we're no longer trying to mimic human intelligence. This is taking the neural network idea, but looking at it really as an engineering or mathematical abstraction. The early ones also had what was just called a single layer. So it would take, pressing the wrong button, I'll try and go back. It would take an input here as shown in red, and it would just pass this right to an output. The modern deep neural networks, the term deep comes from, it stacks many, many different versions here of these computational functions together. So it can compute very, very complicated mathematical functions, but still through this process we call training or machine learning. What's enabled this has been a few different factors. There's been certainly developments in the theory and software, but hardware has been a big thing. Graphical processing units starting out in the gaming industry, but now going into scientific computing have been a huge thing. But perhaps the biggest thing overall has been the availability of lots and lots of data to train these models on. So this has led to a concept called foundation models. And the idea is to train on very large data sets that are often heterogeneous. So this is looking at one that would take medical records, X-ray information, et cetera, and train this into a model, and then to use that to do a variety of different tasks. So this could include things like answering questions about the data, summarizing information, et cetera. So this is a system we developed in the division I oversee at Harvard. that's called Path Chat, updated. This was just accepted in Nature last week, so I didn't have time to update it. But this is a system for pathologists where you can show it an image and you can have a conversation or a chat with the machine. You can ask it questions, what type of cells am I seeing? You can even ask it what to do next. So really this notion of mimicking human intelligence or a pathologist in this type of system. So what are some of the challenges to applying these tools to the microbiome field? So we certainly do not have the amount of data that many of these systems were trained on. When you're looking at systems like ChatGPT, the current version, it was essentially trained on the entire internet. So you're talking about billions and billions of data points. Microbiome studies, and the unit that's really most relevant is the number of people studied, are going to always by nature be much smaller than that. The data also are very noisy. This has to do with the experimental methodologies, but also what we're looking at. People vary in their microbiome a lot, and this has to do with what I call existential variability. You have different microbes than the person sitting next to you. It's also a complicated data set. There are things like evolutionary relationships we have to take into account, so it's not just measuring lots and lots of independent data points. We also do a lot of special experimental designs. A number of people previously have talked about the importance of longitudinal studies and that type of thing, as well as multi-omics, so integrating lots of different views of the microbiome. That's also complicated. And the last one, though, I really want to draw your attention to, it's very important that we're able to interpret what these models are doing. When you look at a system like ChatGPT or image recognition systems, they're essentially black boxes. We don't know or care as much what they're doing, but in a biomedical context, that won't work. And what I'm showing there is a neural network model. This actually predicts for the sort of composition for a concrete or that type of thing, how solid it would be. It's very good at predicting, but there's no way anyone could look at that. It's just a series of numbers, et cetera, these connections. You cannot tell why a particular material composition is best. So what I want to present is a very short kind of case study of work my lab does to try and create purpose-built deep learning models that address these types of challenges. So the setup here is we wanted a general method to be able to look at longitudinal microbiome studies. The setup is we're measuring some group over time, and they perhaps are at risk for a disease, et cetera, and we want to make a prediction. So this could literally be a disease prediction. What is the risk of developing type 1 diabetes or recurrent infection? Or it could be more of what we call a classification thing. We're trying to look at how vaginal versus C-section babies, for instance, differ in terms of some characteristic. So we developed a few years ago a system that we call MITRE for short, or the Microbial Interpretable Temporal Rule Engine for the long term. And what goes into that system are 3 inputs. We have a time series or longitudinal microbiome data. We also take into account the phylogeny or evolutionary relationships in the microbes. And then this is a so-called supervised machine learning task. So we are trying to predict something, so we require information about the host as well. That goes into a piece here that infers from that data. And then what I want to draw your attention to is the output, which I'll go over in the next slides. But these are human interpretable rules that predict whatever the outcome is. And these rules have a very particular structure that we build in. So they consist of what are called detectors. Each detector has 3 different parts. So the first part is it says, I'm going to identify a group of microbes, so a part of this phylogenetic tree that's most relevant to the prediction. It also says I'm going to find a time window. So in this case, it shows 3 to 5 months, and then it looks for a threshold. And from that, it can make a prediction about the disease state. It can build up more complicated rules. So it can chain together these different detectors. Here it says, this first one has to be true, and then a second one, which looks at a different time interval, a different set of microbes, and a different threshold. If both of those things are true, then your disease is such and such. So this can really sort of make fairly complicated predictions, but still you can go back and always understand how the computer made that prediction. So this was our work circa about 2019. Unfortunately, it was too slow. When we started to scale this up to larger studies with hundreds of subjects, it would take days to run. And that's where we started turning to deep learning. Before I go into that, I'll just say the other thing when we do these type of studies, we have to benchmark them. So that what it means is we have to ask how they work against previous or prior methods. Won't go into the details of this, just to say one of the important comparisons is against something called a random forest. This is another deep learning model, a machine learning model. It's been mentioned by other speakers. What's important to understand, that is a black box. You can't directly interpret what it's doing. And what was nice is our model, which is shown here in blue and orange, performs usually as well and sometimes better than that comparative method, but is fully interpretable. Okay, so as I said, that was circa 2019. Too slow. So what we did is said, can we kind of reconfigure this so it works in a deep learning framework? Remember I talked about how the deep learning had these different layers, and it was this idea of these neural networks. I said, well, how do we convert this into that kind of methodology? So what we ended up doing, we invented a lot of new mathematics to do this, but you can view each part of what that previous model did as a layer in this neural network. So there's one that looks at the phylogeny, there's one that selects the time interval, et cetera. And so once it learns this model, it can directly then, you can still read off that same rule. And this model, it runs as about 1,000 times faster, but has the same accuracy as the other model. So here's just sort of a kind of little case study showing you how that works. We also have a graphical user interface. This was a study looking at infants in the first year of life, so certainly relevant to this conference. And these were breastfed versus formula-fed infants. So we asked the model to find rules that would differentiate those infants. In the interest of time, I'm just going to take you through one of those. And that's the one down here, which I think is microscopic. You probably can't read it, but I will just point out to you on the graph what this looks like. So what the model did, interestingly, it selected this time window, which is basically between 3 to 6 months of life. And up here, I think, yeah, the formula-fed is down here, and breastfed is the upper panel. And so what's really interesting, this first period of about 3 months, the microbiome is it's very individualized, almost chaotic. It's hard to tell any group apart that way. And then if you start looking at this 6 months on, that's around when solid food is introduced. And so the microbiome started to become more similar. So the method automatically honed in on this time window of 3 to 6 months. That's when it could differentiate these groups best. And then what did it hone in on in terms of microbes? So this is a group of bugs that they're all Clostridiales, and these tend to be bugs that are better able to metabolize more complex foods. And so you see this kind of earlier maturation in the formula-fed infants, if you want to look at it that way, or in other ways, they have this kind of more complex Clostridiales community earlier. So the model automatically finds all those things without additional needs for someone to identify the time window, the bugs, et cetera. All right, so I want to talk a little bit about sort of future, what may feel like science fiction, but the field is moving so fast, I don't think it will be. So what are some things we'll be able to do with these tools in the future? One of them that's really exciting is to be able to make forecasts and relate the individual level. So to look at a patient and ask if they have a particular microbiome configuration and ask if things happen to them. They had dietary changes, antibiotics, et cetera, to make predictions about what happened to their microbiome and their health status. Another one is really using these tools to delve in to get much more mechanisms, so start to understand the molecular biology, and in particular, how is the host and microbiome interacting. A third one, which is really dovetails nicely with the last talk, is starting to understand what I would call the metagenomic dark matter. So there are millions of microbial genes, we don't know their function. You add bacteria, and then on top of that you add viruses. It's a vast universe of things. And these tools really have a capability to start unlocking that. Methods like AlphaFold, et cetera, where you can start predicting from sequence the protein structure. A huge amount of potential. Another one is to be able to start looking at the spatial structure of the microbiome, mapping out how microbes are talking to each other within the gut, how they're talking to the host within the gut spatially. Again, a huge potential. And then the final one, which we're already sort of started seeing pieces of, I showed it with this PATH chat thing, but I believe we could do this for the microbiome, is to really have an AI system that would be capable of having a more understanding of the literature, existing mechanisms, et cetera. You could have a conversation with it. You find something, you can ask it, what is it, what do I do next, et cetera. So having that AI kind of in our study design experimental loop. One I don't put on there, but we just say to this room, I think also clinically many of these tools are coming very soon with similar capabilities. With all that potential, I do want to say something about the dangers and sort of caveats here. So there have been a number of studies already showing that there is bias baked into a lot of these large deep learning systems. And a lot of that has to do with the data they were trained on. So if there are underrepresented populations that weren't in the training data, those are not going to be well represented in what the system outputs. So it's really critical to start thinking about how we can reduce that bias in these systems. There's also a real danger of removing humans from judgment and creativity. These systems, ChatGPT is a statistical model. It predicts what word is most likely to come next. It does not have human judgment. It does not understand what's going on. It appears very intelligent. People are very honed in. When something's using language, it appears smart. It isn't. And we have to really think of it that way. Another risk is these systems are largely being developed by large and mostly American companies. And their agenda may not be the same as, say, global world health, these sorts of issues. And so we need to look carefully at who is inventing these technologies, who is controlling them, et cetera. There's also a negative impact on the environment. It's been stated that every time you generate an image on one of these new image generators, it is amount of energy equivalent to about boiling a bottle of water like the ones in front of you. So there is really a need to think more carefully about energy use, environmental impact of these potentially powerful tools. So my kind of red caution tape thing is I think that incredibly powerful tools, but there's a real need for the people creating them to be accountable and engage with all stakeholders to chart a course for responsible use. So just some key takeaways. AI is a field that has been around for about 50 years now, but there are some recent advances leading to a so-called deep learning revolution. And I can say I really do think it is a revolution. I've been in the field, as I said, more than 30 years. I've never seen this acceleration and capability. So I think it's here. We have to deal with it. It's very powerful. From the microbiome perspective, there are specific challenges that I mentioned, particularly for just applying these off-the-shelf systems. But I think there's a good opportunity to build specific tools for the microbiome field that will really enable some very, very exciting applications to move things forward, even in the next few years. But what I talked about in the last slide and emphasize again, there are some real dangers in AI. I think this is going to take international efforts. It's going to need to be combined public interest, academic, and corporate, because they do control a lot of the technology, to use these tools responsibly. All right.
Thank you, Georg. Please have a seat. And I invite all the others, please. Also, yeah, Andrew. Yeah. Thank you very much. This was exactly what we wanted, to have a look into the future. Thanks a lot.
Thank you.
So we invite again everyone here in the audience and also everyone who is joining us online. So please submit your questions. The idea of the session was to have a look into the future. What are the future directions? What are the trends? What will happen at the next meeting? Flavia, do we have some questions from the...
Yeah, the problem is that it's not working.
Oh, it's not working.
This mine, maybe, yeah.
So perhaps we can start the discussion and perhaps you can open the...
Yes.
So Georg, can I start with you? Because you started with artificial intelligence. I'm not saying that other questions that other topics are not equally fascinating. All of them are fascinating because that's why we have them here. But with AI, which is on the rise, and there is an increasing number of papers on artificial intelligence, machine learning, but not so many of these research achievements have been already translated into healthcare. So what could be the reason behind it? Why so few of them have been really used in healthcare? What do you think?
Well, I mean, I think a lot of it comes down to the economics and the priorities of, where it is stressed.
Can you
hear me, or is it? Okay.
Yeah.
I think many of the early applications have been in the commercial sector. If you look at where they're trying to apply it now, it's things like office assistants, in entertainment, et cetera. So I don't think it's necessarily the capability isn't there, but I think there hasn't been the emphasis on it. I think there is also which is a good thing, a little more degree of caution in the healthcare space with just rolling these tools out willy-nilly. So
and who will be responsible for the final decision eventually? Because we really don't want to make AI to make the decisions. I would prefer to have a human. You mentioned black box, and I fully agree with you that there's a lot of misunderstanding, we do not understand. On the other hand, there is one thing, in medicine, there are some interventions we also don't know how they work. And they usually use one example, which is fecal microbiota transplantation, extremely effective for recurrent Clostridioides difficile. But we don't know really how does it work. So it's not that it's only with artificial intelligence.
No, that's very true. I mean, when I trained in medicine, there's actually a lot of things. We don't understand the mechanism of action.
It's important that we do.
It's important to do, but they work clinically, and that it can't be disregarded, right?
I think that Flavia managed to open.
Yeah. There is a lot of questions. I'll start with this first one that was 1 hour ago for Dr. Andrew. Why would you have your microbiome mapped? So this is the question. Can we responsibly translate this as a biomarker of disease? I mean, he asked, what's the meaning? Why do we have to mark our microbiome?
So there are a variety of people that are interested in diet, nutrition, and how those things manifest within their bodies. So a lot of the people that come to the Microseta initiative are in that group. Why we map it, we don't necessarily have the evidence to relate someone's microbiome to a disease state.
Okay.
The data isn't there for that type of assessment. What we do is we relate someone's microbiome to another meta-category. For instance, how closely an individual is from a Mediterranean diet or a vegetarian diet, or how closely their microbiome maps to some other aspect of their health and wellness.
Yeah.
That's why we map it.
Thank you.
It's not an absolute.
Yeah. So another question for you again. Could you elaborate the main goals with this THDMI project? Is fecal metabolomics also explored? And how deep is the clinical and nutrition level data collected?
So how is the metabolomics? So currently we're not collecting metabolomics data from the samples that were part of that study.
Okay.
That's the easy answer to that first question. The samples are self-harvested, so there isn't a clinical component to that collection effort, so that a clinician is not involved. In some of our partners, we have had where we have had a clinical input into as more of a convenience for our users to come to a central location to have their sample collected, but the intent is for them to self-harvest their sample.
Okay, thank you. Now this is a question for Sasha. So for Dr. Sasha, do you have any thoughts on the consequences behind the clear difference in temperate and lytic phage concentration in between infant and adult gut?
I think that this is just the mechanism how the phages are appearing in the gut. So most bacteriophages that we see in babies are those that are coming from bacteria. It's a different condition. Bacteria are stressed because they are located in a different environment, and then they start to produce bacteriophages. After a while, it becomes a different ecosystem, it's settled down, and then we do see the different proportion more similar to adults.
Yeah, just I continue with you. Another question is, does viral abundance benefit our health or opposite?
I don't think we can say that. It's of course very much related to the bacterial abundances. So, because if you have a lot of hosts, then you also have a lot of viruses. We try to correct for that. We try to look for viruses separately. It's very difficult. We need big numbers. We need AI maybe for that.
Yeah.
But I would not say that we know if the viral abundances is a good or bad thing.
Okay. Now back to Andrew. So does health quality is actually better within less urbanized population? In the means, does this more diverse microbiome result in a better health?
There's a general sentiment that a greater biodiversity in your gut is related to having better health. And that also goes to the diversity of the types of foods that are consumed. So the broader the distribution of food types that you eat, generally there is an association with better health.
I'm just looking around if there's no one from the audience.
Francesco De Seta. Francesco?
Yes, please. Because we should give equal rights to online.
There's a lot of questions coming.
Yeah, online and audience and those present here.
Just a question for artificial intelligence. We made a big mistake in the last 50 years regarding gender medicine. Is there gender bias in artificial intelligence? Because some data, they report that we start just to study men and no women. I'm a gynecologist, so sorry.
White man.
So the short answer is absolutely. You know, much of this bias comes from how the systems were trained, so what data they saw, and so you have lots of studies that yes, it's white males, and that's what they're seeing. There's also other more subtle sources of bias though that are quite disturbing. You know, there was a system that Amazon had for employment screening, and it was systematically discriminating against female applicants because Amazon had systematically discriminated against them during employment. And so it basically, it learns to do what we tell it. And, you know, to their credit, they found this, it was not deployed and it didn't cause harm. But that's the sort of thing, yes, people need to be very cognizant of translating the bias in studies or cognitive biases in people training the models.
If I can go on this. If there are any other questions from the audience? No? So I can go again to Andrew. How long does it take for a gut microbiome to adjust to favor to a new environment? In example, migration, traveling?
That's a great question. I think it depends. When we do interventional types of trials, we usually have a washout period that's measured in weeks to months. So if that's any indication of an intervention versus a non-intervention, I think that you should likely see recovery within weeks to months of your microbiome.
Okay.
We have more questions for Sasha, Professor Sasha. So what do you think about the possible interactions between phages and mobile genetic elements like plasmids and how they influence the infant gut?
I think that I think we do not know much about that. We also see plasmids in our data set. What we now do is setting them apart and waiting for the analysis of them separately from bacteriophages. But of course, it's all very much related ecosystem. Yes, phages and plasmids. And there were a few publications about the role of plasmids and how they can transfer genes, in particular antibiotic resistance genes is one of the groups that people are usually interested in. So they are important, but how exactly they are related to viruses, I think is difficult to say. Plasmids are usually more abundant and more similar. There are some plasmids that are very common in the human gut.
Yeah. Sasha, now when we are starting about discussing this gut virome, why it's so that we were interested for such a long time in bacteria? We knew there are viruses, and there was hardly any interest. So what were the triggers that really changed the direction of interest? Is the methods which are now?
I think we need to look for more things in the gut. Yes, it's not only viruses that are now getting interested. It's also, of course, microbiome.
Exactly.
It's also protozoa. It's archaea. I think these are all important. I would not really say that viruses are more important than bacteria, but historically we did have the 16S method, which is a great method. It's a marker gene for bacteria, and viruses, they do not have marker genes, at least DNA viruses. You cannot really catch viruses as easily as you could have catched the bacteria.
I also have another question because we have many young people in the audience. Some of them are probably thinking about doing some research. In your area, in this gut virome, what would you think is like the next step to evaluate, to assess?
Oh, you can really go all directions there because we
Just
are
give us some top directions.
Yeah, I think everyone have some preferences for their research interest. I think for me, maybe also because I came from pediatrics and from immune diseases, I am very, very fascinated by the immune system of bacteria against viruses. It's so complex, it's so fine-tuned. We really have to learn from our bacteria and viruses how they interact with each other for setting up new immune developments in host.
And AI that we are discussing today, what is the role for? What do you think?
I think it will be a magic, but I think we need to, of course, to think how we apply that, because I see a lot of opportunities, but also a lot of challenges. But I think that these questions should come to the expert.
I could say one thing about viruses in particular that's interesting. So your comment as to why they weren't studied earlier, I think is right, spot on. They're so diverse that there was no marker gene, and it took the technologies to catch up. Similarly with the bioinformatics, that's one of the problems. Looking at the sequence level, there is so much variation. A lot of the tools that had worked in the past don't. One of the capabilities with AI is to be able to predict the structure, because that's a lot more conserved. So things like capsid or even enzymes. And so I think that's going to be a pretty immediate application, is to much better characterize the viruses by predicting from their sequence what the virus structure actually is.
Flavia, your turn.
Yeah, thank you. So I go for this to Sasha again. So many bacteriophages are inserted into the bacterial genome. Can we study them analyzing the bacterial genome sequences?
Yes, that's what we can do. It is one of the way of looking for viruses. We do see bacteriophages there. What we can also do, actually, that's some work that is ongoing in the lab and in other labs as well. We can induce these prophages. We can take cultures of bacteria that are available in the lab. We can stimulate these phages with everything, light, temperature, chemicals, and then see how they are being produced and how they actually look like. Because the problem is that we sometimes do not know how they exactly look, how they function, what kind of genes they have. And this is the way of studying them.
Yeah. So this question is for Georg. Say that this is something that maybe we need to discuss a little bit. Say this is also important to say that artificial intelligence is not intelligent because it's a coding created from humans, by humans. So maybe you can tell us a little more about controlled, not controlled, or guided, not guided, artificial intelligence algorithm. Yeah.
Yeah. So I think since the start of the field, this has always been a question: Are these systems intelligent? What constitutes intelligence? And I think for computer scientists, that really take an operational view that it is mimicking behavior that it appears to be intelligent. Attributing actual intelligence is a whole other thing that I think I personally would be very cautious to do. And it's really important. I mentioned that about ChatGPT, but it's really important with these systems to think about this, because the human brain is almost tuned to, there's certain behaviors we see and we attribute that to intelligence or human-like. Use of language, ability to generate pictures that look coherent, et cetera. So we read a lot into these systems about what their capabilities are. In the machine learning field, though, we try to be very objective. So what is its actual capability? What is it doing, et cetera? And that's more how I personally would look at it and think about defining it. Another kind of part of that question, I think, is how controllable are these systems? And I think that is really a key thing. The current generation of them, again, like ChatGPT, are not very controllable. They were trained on lots of data. They just output what is predicted to be the most likely next thing. The artistic systems, in many ways, I think are awful because there's no intention to what they produce. So there's a huge need for this to make the systems more controllable, also to make sure that humans are in the loop with the decision making. And I think that is going to be hopefully the next generation of systems we see.
Georg, I have some question to you regarding artificial intelligence. We discussed it previously that there is among healthcare professionals somehow like a feeling of a threat when it comes to artificial intelligence, and there are variation in approaches from fear, from skepticism to enthusiasm, or even over enthusiasm. But let's focus on those who are really feared of or scared of artificial intelligence. How we can overcome. It's not only us professionals, but it's also for patients, because they are also scared of using artificial intelligence for decision making. So what we can do, all of us, like healthcare professionals, in order to overcome all this fear?
Yeah, I think it's good to have some fear or skepticism of these systems, and when communicating to a patient, I think at least, hopefully forever, but in the current regime, to emphasize these tools are being used not to make the decision. It's equivalent to, if you had a reference where you were looking something up, or you had an assistant who were providing some information to you, but it is not that the artificial intelligence is going to be making a clinical decision. There are circumstances where that is already not the case. It's starting to happen. There have been FDA-approved diagnostic systems, et cetera. Again, there is still going to be, at present time, a physician in the loop with that. But how that will be in the future, I think it's something we do as healthcare professionals need to advocate and not let, at times, the commercial interest run away with, okay, there's a potential here, but there are very serious safety issues, there are very serious trust issues with just letting that run.
Yeah, this is the question for Andrew. If interracial marriage may lead to heterogeneity in gene pool for future generation, will this also make our microbiomes more diverse? I mean, with this interracial marriage, and should their effect subdue their effect?
I don't think that there's evidence for anything like that, so I don't know how to answer that.
Okay. So then again, Sasha, you have data about the effect of COVID on children virome pattern.
We didn't analyze it in line with that yet. So this data set was collecting before, during, and after COVID, but the data freeze that we have been analyzing so far, it is before and during COVID. So what we are planning to do is to finish the data collection and then to have a look. We do see, of course, a huge effect of COVID on also all kind of behavior in the Dutch population.
And there is coming the question related to this and that question. There is some evidence with persistent SARS-CoV-19 in the gut of the primary infection without vaccination, some association with the long COVID infection? Well, long COVID symptoms.
In kids, we do not really, in these kids, we do not have these long COVID symptoms. We cannot really say anything about that. Yes, the COVID, the virus, the SARS virus is identified in stool. It's not very clear how it is there. Maybe it infects the intestinal cells.
We have a question from the audience.
Hi.
Please.
Can you hear me? Okay, so Vidya Ramchal Tamaraj, Trinidad and Tobago. And this was to sort of comment also on the question that you just asked about the artificial intelligence and doctors. Because China has unveiled their world's first AI hospital with 14 AI doctors. So they are definitely moving towards that. And that's also because of their population being very aged and a lack of caregivers. The fact is, it's a technology acceptance model that we will have to look at. And they have basically stated it's not to replace doctors, but it is more to augment. And it is a fair factor in that most doctors think they're going to be replaced, but it's also the perception of the population as to how they will accept it. And I think it's also culture-based. A lot of people prefer more doctor-personal interactions, and that's something that will have to be looked at in the future, as well as how you said the AI has to be controlled to a certain extent because it has definitely mushroomed beyond, and we have to look at it as more how it helps us than to fear it in that sense.
Yeah, I mean, I think that's hopefully where it will go is that it's augmenting the healthcare system. And, you know, we have similar problems in the U.S., actually. There are states with very underserved, particularly with pediatricians or specialists. And so that's a lot of where people are looking at this right now is could it augment, could it make certain tasks go faster? So yeah, again, I think there is a huge potential good with the technology. It's just going to need really thoughtful application.
So there is this question by Dr. Oluwole Banjo from Nigeria. So he says, I've seen data from various parts of the world in these works. Do we have data from the global south input into all this study, especially for artificial intelligence study? Do we have study from this southern part of the world? This is for all of three speakers.
What is south part of the world? Actually, there are
Let's say Africa.
Yeah, because there are some beautiful.
Let's say Africa.
Quite some beautiful research in New Zealand in terms of viruses, but I don't think it answered the question.
No.
I think that for the early life microbiome cohorts, there are of course cohorts, for example, from South Africa. The virus-specific analysis, it started in the north, but it definitely will go there as well. So that's, I think, from this side.
I mean, I think those areas of the world, from what I've seen, are underrepresented in a lot of the microbiome studies. I think that is a gap that needs to be worked on.
Yeah, I'm aware of some studies. I would, I guess, characterize them as being very focused studies for data collection throughout the Southern Hemisphere. But it's not for the lack of programs or effort.
So, thank you. And so this is for Sasha. Is there any potential danger in considering the use of viruses as therapeutical option? Aren't viruses capable of mutating into dangerous agents?
Viruses can be dangerous, yes. That's what we have learned. I think all of us almost became a virologist in the past few years. So the potential answer is yes. On the other side, we know that, for example, breast milk, it has viruses, and we know that it is not dangerous for the baby at all. So I think viruses, there are different viruses, and they should be really discriminated and very, very well studied before we use them as a therapeutic potential. That's for sure.
Yeah. And this is for Georg. So are there studies that use the mother's data with artificial intelligence to predict the outcome of the baby pregnancy or health?
I'm not aware of, but there could well be. Again, it goes back to collecting sufficient data to power these studies or be able to do that. But it's an interesting area. I'm not aware of those studies, though.
Flavia, there's another question for Georg. Do you encourage other groups to use your microbiome AI tools in the research? Is a question from PhD student.
Yes, we absolutely do. So in my group, everything is released as open source, is free and fully documented. And I would just, one more thing to add, what I was mentioning about these latest protein folding fluids. AlphaFold 3 had the unique distinction when they published that in, I believe it was Science or maybe it was Nature, they did not publish the source code. They didn't even publish the full method. So the openness of this, I think, is an important thing, and we're all in on that.
It's also for you, this Dr. Claudia Martinez, she wants to collaborate, and she say, how can we collaborate for your study? So I think that she can contact you. Yeah. Your email, it's free. And also another question for Georg. I don't know what is, maybe you can help, I don't know. I don't understand this question. Is what the danger of malicious software? I don't know what is malicious software.
What was the first part of the question?
Malicious software.
I know about malicious software.
And say,
what are the dangers?
what is the danger?
Okay. Yeah, so I think there's a few things about that. I mean, this has already occurred in some of these systems. There's various ways that they can be malicious. Traditional software, we had things like virus, not a real virus, but a computer virus and what were called worms and other ways. One of the ways, though, that's new with these things like ChatGPT is they can present misinformation, and sometimes this appears to be, at least gives the appearance almost to be intentional. And it's a big danger, particularly in these sorts of healthcare applications, because when either a physician relying on this or patients directly interacting with these systems, the concept that the information has to be reliable, also going back to your presentation very much, it needs to focus on randomized controlled studies. It needs to focus on quality evidence. That's what it's reporting, not just random things it read on Reddit on the internet. So I think that's a real gap in this application that needs to be worked on strongly.
So I think this is the last, if we have some couple of minutes. So artificial intelligence will be great if we can, can be properly integrated into medicine, especially in biome research. But how do you ensure artificial intelligence will be acceptable by parents?
So if going back to these exceptions,
going back,
by healthcare professionals, but
exactly.
Also by
so
patients.
How can we say to the parents, look, we are using this?
Yeah, I mean, one thing I would comment, so a person in my group used to work on aircraft, so worked at Boeing, and I think there are models for safety here. I mean, when you fly in an airplane, frankly, it's kind of scary. You're relying on this going to the sky, does it even work? There are models for very advanced technologies being accepted by the public, so I think we need to think in terms of those types of models. I agree. I think for parents, handing a baby over to this AI system, it's got to have all those safety things in place, et cetera. So I think there's certification, there's controllability, all those things will need to really go into this before it's widely deployed.
And we did not discuss the whole area of ethical issues which are related with AI. I'm afraid we will not have time because we have only 30 seconds. So thanks a lot. Thank you very much. I think that it's obvious that science never solves Many thanks to all the speakers, that the science never solves a problem without creating 10 more, if even not more. So thanks a lot. There is a lot of opportunities for further research, and I do hope we will see a lot of new advancement very soon. Thanks a lot. And many thanks to NNI for organizing this session, inspiring session. Thanks a lot. Thank you very much.